Today, our topic is Vectors in Tools for Genetic Engineering, Principles and Process of Biotechnology, Unit 8, Chapter 4 in Botany. So, what is a vector? Have you heard this vector? So, you can know this vector algebra and you can hear this word in maths. But here also you can hear this word vector in diseases. That is, there are some organisms which causes disease. For that organism, they need an transport agent known as vector. For example, malaria was caused by mosquito, you are all thinking, but malaria was caused by protozoan plasmodium. This plasmodium protozoan was carried by mosquito. So, carried by mosquito. If that mosquito injects a person, the person get malaria. So, here the malaria is that is the mosquito, not the malaria. The mosquito acting as a carrier. So, it is called as vector. So, the same meaning only here also vector. But here we are going to use for the genetic engineering purpose of carrying the genes for altering to make our DNA. So, for that first we will see vectors. Another major component of a gene cloning experiment is a vector such as plasmid. So, for gene cloning or rDNA technology or genetic engineering, the major component is vector. Here the vector is plasmid. We are using the plasmid as vector. A vector is a simple DNA molecule capable of self-replication and is used as a carrier and transporter of DNA fragment which is inserted into it for cloning experiments. So, vector is a small DNA molecule only and it can be able to replicate by itself, by its own. So, self-replication means the vector can be able to replicate by its own and small DNA molecule, and it is acting as a carrier to carry the gene of interest. No, so carrier and a transporter of DNA fragment. So, carrier and transport to another organism. No, so that is a transporter carrier which is inserted into it for cloning experiments. So, the DNA fragments are inserted in it in the vector, and this vector acting as a carrier and transporter for what? For the cloning experiment. Okay. So, vector is also called cloning vehicle or cloning DNA. So, the vector is a cloning vehicle because it taking the gene to which the gene going to be cloned. Okay. So, we can name it as cloning vehicle. So, it is acting as a vehicle which travels from one place to another place. So, it travels from one place to another place. Okay. Because of that, it is called cloning vehicle or cloning DNA. So, the vectors are of two types, cloning vector and expressed vector. So, the cloning vector is used for the cloning of DNA insert inside the suitable host cell. So, the cloning vector is helpful for the DNA insert to clone. So, only to the DNA insert which going to be inserted into the vector, that one has to be cloned. For that purpose, used vectors are called cloning vectors. And expressed vectors used to express the DNA insert for producing specific protein inside the host. So, the DNA, that is expressed vectors, it is used for the DNA insert. So, we are insert the DNA and it has to produce the specific protein inside the host. For that, they are called expressing. They are expressing another character. Cloning vector, only the DNA insert has to clone inside the host cell. Okay. But in expressed vector, along with the DNA that is insert, that has to be cloned. The DNA insert and as well as the specific protein has to be cloned in the host. That is called expressed vectors. Now, as we are talking about the plasmids, so this is a bacterial cell and you can see the circular DNA particle. They are the self-replicating one which is called plasmid, and this is the bacterial chromosome. We are talking about this plasmids which act as a vectors. Properties of vectors. So the vectors properties. So to replicate autonomously, to produce multiple copies of them along with the DNA insert in the host cell. So to replicate means autonomously means, as we say, it replicates of its own. So replication, DNA replication take by itself. It doesn't depend on the bacterial chromosome. So this is the main DNA. It doesn't depend on it. So it can replicate by its own. That is called autonomously. To produce, why it autonomously replicating? To produce multiple copies of them along with their DNA insert in the host cell. So it replicates by its own and it produces more copies of DNA insert along with the host. Okay. It should be small in size and of low molecular weight, less than 10 kb in size, so that the entry or transfer into the host cell is easy. So the vector should be in small size. Okay, it should be in small size and the molecular weight should be low. And how much? 10 kilo base pairs. Kb means kilo base pairs. 10 kilo base pairs. Not more than that. It should be less than 10 kilo base pairs. So keep it in mind. This they may ask in 1 mark and in your NEET exam. Why it should be in small size and low molecular weight and less than 10 kb? Why means it is easy to transfer. Okay, easy to transfer or enter into the host cell easily. So the vector must contain an origin of replication so it can independently replicate within the host. So this vector DNA has an origin of replication, ORI. So origin of replication means if the origin of replication is present in a DNA, it can replicate by its own. It cannot depend on the host DNA to replicate. So it can replicate by its own. So because of that, the vector should possess ORI, that is origin of replication. It should contain a suitable marker such as antibiotic resistance to permit its detection in transformed host cell. So it should contain first ORI. The next one is suitable marker. Why the suitable marker? It has to find out the antibiotic resistance. Why? To detect the transformed host cell. So which is a transformed host cell? That is the changed, the plasmid, that is cloned vector is which is the vector which carries the gene of interest, that it has to find out. Because of that, it needs a suitable marker. The vector should have unique target size for integration with DNA insert and should have the ability to integrate with DNA insert. So it has a unique target size where the DNA insert going to join with the vector. So the particular site only it has the DNA interest or DNA insert can join. So that site should be there. That integration means joining, and show how the ability to integrate with DNA insert. So first there should be a unique site for joining the DNA insert with vector, and it has the ability to. So for joining site should be there and it has the ability to join the DNA insert. Okay. It carries into the genome of the host cell. So it carries, why it has to be integrate? It carries the genome of the host cell also. Because of that, it has the unique site and ability to join with the DNA insert. Most of the commonly used cloning vectors have more than one restriction site. So the vectors you can see here. So you can see this is the restriction sites. So a vector, this is a vector which has more than one restriction site. Okay. So commonly they have more than one restriction sites. So these are the multiple cloning site or polylinker. So the restriction sites, so this restriction sites are considered as polylinker or MCS, multiple cloning sites. So presence of multiple cloning sites facilitates the use of restriction enzymes of choice. So you have so many MCS, that is multiple cloning sites. Because of that, the vector has select different enzymes. So many restriction enzymes are there. Which enzyme it needs to work here, that can be selected by the vector. And these are the properties of vector, and the following are the features that are required to facilitate the cloning into a vector. So the cloning, the facilitate, that is the required to facilitate the cloning into a vector. So first you need a ORI, that is origin of replication. So the vector must contain an origin of replication recognition in the host cell so that it is replicate along with the DNA that it carries. So it need a replication site, that is ORI. Y means it carries the DNA site. When it enter into the host cell, it has to replicate along with the DNA it carries. For that, it need a ORI. Next, the suitor, that is selectable marker. It should carry a selectable marker trait that enables the cell containing the vector to be selected or identified. So why it carries the selectable marker, in which it has going to be joined with the insert, DNA insert, and to identify the DNA insert and to identify the cloning DNA also. For that reason, it carries the selectable marker. So this is the selectable marker. And the third point is, a cloning vector needs a single cleavage site for each of one or more restriction enzymes. So the cloning vector should need a single cleavage, means cut. So cut for each restriction enzyme. HIND3 is here, EcoRI, SATI, BAMHI and PST. So these are the restriction enzyme, and each one has a single cut on the particular vector DNA. Okay. So now we will see the features to facilitate the vector. Okay. First is the origin of replication, ORI. So this is a sequence from where the replication starts, and the piece of DNA when linked to this sequence can be made to replicate within the host cell. So ORI, this is the ORI site. The origin of replication should be there in the cloning vector, and where the DNA join, that is insert join, also it has to replicate, and here the sequence starts to replicate. Okay. Next, selectable marker. In addition to one vector, requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformant. So, the suitable marker will identify and select the transformed DNA as well as non-transformed DNA to remove, to add on to identify the transformed DNA and to replicate it, and the non-transformed DNA to eliminate. For this purpose, the selectable marker are taken. And cloning sites. In order to link the alien DNA is the DNA of foreign DNA also we can say. Foreign DNA, the DNA of another organism like that. Okay. So, in order to link the alien DNA, the vector need to have few preferable single recognition sites for the commonly used enzymes as we had said already. So, that is for the foreign DNA to cleave the restriction site, that is in the restriction enzyme need a single restriction site to cut. Okay. So, that is called the cloning site. Okay. Types of vectors. Plasmids. Plasmids are the extra chromosomal, self-replicating, circular DNA found in the bacterial cell in addition to the bacterial chromosomes. So, this is the extra chromosomal ring which is circular addition to the bacterial chromosomal DNA. It is present in the bacteria. It is called plasmid. So, the plasmid contains genetic information for their own replication, and plasmid is an extra chromosomal. So, it has an DNA which is, that is it has a separate DNA and it can able to replicate by its own. So, it is seen in the bacterial cell and it is an additional one in bacteria because bacteria has the bacterial chromosomes also. PBr322 plasmid. PBr322 plasmid is a reconstructed plasmid and most widely used as a cloning vector. So, it is a reconstructed plasmid which is done in the laboratory. Okay, and it is widely used cloning vector and it contains 4361 base pair. This they may ask in your 1 mark as well as in your NEET exam. So, in PBr, P denotes plasmid. Okay, P denotes plasmid and capital B and R denotes the scientist Bolivar and Rodriguez. Who developed this plasmid. And the number 322, the number of plasmids developed from the laboratory. So, in the laboratory, this plasmid was developed 322 number is the number of plasmid has developed and this number is PBr 322 is developed. It contains an AMPR and TET-R, two different antibiotic resistant. AMP is ampicillin resistant and TETR is tetracycline resistant genes. And recognition sites for several restriction enzymes like HIND3, EcoRI, BAMH1, HI, SAT-I, PVU2, PST1, that is PSTI, CLAI on the antibiotic resistant genes and rope codes for the proteins involved in the replication of plasmid. So, PBr322 plasmid is an important plasmid. They may ask in your 3 mark as well as in your 5 mark. So, it is reconstructed plasmid and it is widely used as cloning vector. It contains 4361 base pairs and PBr, P denotes plasmid, B and R denotes, B denotes Bolivar and R is Rodriguez. They have developed the plasmid and number 322 is the number of plasmid developed in the laboratory. This PBr contains AMP R and TET-R. So, they have the resistant, antibiotic resistant, ampicillin resistant gene and tetracycline resistant gene. And they have the ORI, origin of replication, and the restriction sites for the enzymes EcoRI, HIND3 and BAMHI. These are the restriction enzyme sites and PU2 is also. And it has additionally the rope which codes for proteins for which is involved in the replication of plasmid. TI plasmid bacteria. So TI plasmid is found in the cytokinin auxin opine tDNA region and virulence Agrobacterium tumescens, a bacteria responsible for inducing tumors in some of the dicot plants. So the TI plasmid that is contains auxin, cytokinin and opine and have a virulent region and an origin of replication. This TI plasmid is isolated from Agrobacterium tumescens, a soil bacteria which creates tumors in some of the dicot plants like brinjal, tomato, cotton, etc. The plasmid carries transfer, that is TRA gene, which helps to transfer the tDNA from one bacteria to another bacteria or plant cell. So the plasmid has TRA gene. So they have not given here. So this plasmid has a TRA, that is transfer gene. So because of this TRA gene, it can transfer the plasmid, that is it can transfer from one bacteria to another bacteria or the plant cell. It has ONC gene for oncogenicity. ONC means oncogenicity. Oncogenicity means what? Oncogeny. Oncogeny means cancer causing, because it causes tumor in the plants. No. So that gene was there here in this plasmid. ONC gene is for oncogenicity. ORI is for origin of replication and INC for incompatibility. So incompatibility, it will not compatible, that is it will not comparable, it is not a comparable with any of that. So it has an incompatible gene also, INC. The tDNA of TI plasmid is a stably integrated with the plant DNA. So the TI plasmid is joined with the plant DNA. So plant DNA, it is, that is tDNA was joined with effectively and it is stabled with plant DNA. So Agrobacterium plasmids have been used for introduction of genes of desirable traits in the plants. So this Agrobacterium, that is TI plasmids, which is taken from the Agrobacterium, is used for the introduction of genes with the desirable traits like vigored varieties, disease resistant varieties, yield is high, like that desirable traits. For that reason they are inserting this TI plasmids in the plants. So if they ask the TI plasmid bacteria where the TI plasmid has cytokinin, auxin and opine, that is opine tDNA region, opine tDNA region and virulence region, and this TI plasmid was taken from Agrobacterium tumescens, a bacteria which causes tumor disease in some of the plants. So the plasmid contains, what are the genes? You can transfer gene, TRA gene. So what is the function of transfer? So transfer the tDNA from one bacteria to another bacteria or plant cell. ONC gene is for oncogenicity, that is tumor causing. Oncogenicity is a cancer causing. By here we called it as here tumor causing. Okay, gene is there, and ORI gene is the origin of replication, to replicate along with the gene of interest. Because of that, each and every vector should has ORI region. So here it is ORI gene is there, and INC gene, it cannot be comparable with any one of, so incompatibility gene, and tDNA is effectively joined and it is stabled with the plant DNA. Okay, so Agrobacterium is used for the introduction of desirable introduction of genes for the desirable characters in the plants. Okay, so you can see the tDNA region here and ORI region and virulent region, auxin, cytokinin and opine region. So this is the structure of tDNA. If they ask the PBR, you have to write the points and you have to give the diagram for PBR plasmid, and TI plasmid you have to Ti plasmid or tDNA, you have to give the points and you have to write this question. So, hope you understand. If you have any doubts, post your doubts in the comment box. Give a thumbs up, share and subscribe to Science Easy Tech channel.